Hello, everyone. Welcome to today's live broadcast, Advances in Modeling Human Diseases Using Induced Pluripotent Stem Cell-Derived Cellular Models. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Cellular Dynamics. Cellular Dynamics International, CDI, a Fujifilm company, is a leading developer and manufacturer of human cells used in drug discovery, toxicity testing, stem cells banking, and cell therapy development. The company partners with innovators from around the world to combine biologically relevant human cells with the newest technologies to drive advancements in medicine and healthier living. CDI's technology offers the potential to create induced pluripotent stem cells, iPSCs, from anyone, starting with a standard blood draw and followed by the powerful capability to develop into virtually any cell type in the human body. Our proprietary manufacturing system produces billions of cells daily, resulting in inventoried iCell products and donor-specific MyCell products in the quantity, quality, purity, and reproducibility required for drug and cell therapy development. Founded in 2004 by Dr. James Thompson, a pioneer in human pluripotent stem cell research. For more information, please visit cellulardynamics.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would like to introduce today's speakers, Dr. Kile Magnan and Dr. T.K. Feaster. Dr. Kile Magnan, an application scientist at CDI, achieved a bachelor's degree in both physiological psychology and philosophy, followed by a PhD in neuroscience from the University of Wisconsin-Madison. Dr. Magnan worked previously with Kimberly-Clark Corp, Rush Medical Center, and the University of Wisconsin School of Medicine. At CDI, his work includes all aspects of CDI's neuronal products, including electrophysiological behavior and measurements. Dr. Magnan's evaluation on neural network integration and manufacturing with the iPSC-derived neuronal cultures has helped push the field of MEA analysis and pharmacological screening forward to disease in a dish assays. I'll now turn it over to Dr. Magnan for his presentation.
Hello, thank you for that introduction, Robert, and thank you to CDI and LabRoots in general for organizing this webinar. As Robert just said, I'm Dr. Kile Magnan, and I will be presenting the first part of this webinar titled Advances in Modeling Human Diseases Using Induced Pluripotent Stem Cell-Derived Cellular Models. I will begin by introducing Cellular Dynamics International, CDI. CDI is a company that leverages iPSC technology to manufacture human cells. Induced pluripotent stem cells, iPSCs, provide a platform to increase our understanding of the etiology of human disease states. CDI's technology highlights how iPSCs can help springboard new drug development and therapies via the disease in a dish approach. We will present interesting data from disease states produced by genetically engineered single gene mutations as well as via innate donor samples. CDI has optimized differentiation protocols that convert iPSCs into many different cell types, including, as we'll present here today, neurons, dopaneurons, and cardiomyocytes, but also astrocytes, motor neurons, hepatocytes, macrophages, and the list goes on and on. Obviously, you can visit our website, cellulardynamics.com, and see what kind of cell types we can make for you. As mentioned, there will be two parts to this webinar. I will talk first about neuronal disease models and then turn the microphone over, and Dr. Feaster will discuss some cardiomyocyte disease models. So in this first part of the webinar, I will discuss some of CDI's neuronal cell types, including I-cell neurons and I-cell dopaneurons. My agenda includes presenting data on multiple disease modeling approaches: Alzheimer's disease, Parkinson's disease, and epilepsy. I will first, however, describe our iCell neuron product, which is what we use to put the mutations into and assess as far as neuronal cultures go. iCell neurons are human iPS cells expanded under defined feeder-free conditions and cultured in neuronal differentiation media, followed with neuronal maturation media to induce differentiated neurons that are then cryopreserved after 28 to 30 days total in culture. Neuronal cells produced using this method display appropriate cortical gene markers, including DACH1, FOXG1, and OTX2. The resulting post-thaw of these cells yields a purity of greater than or equal to 85% cortical neurons measured by high expression of beta-3 tubulin and negative expression of nestin by flow cytometry. Pure cells are thus achieved without the need to sort. And this population of neurons can further be defined and tested for synaptic excitatory or inhibitory markers, including B-GLUT2 and VGAT, as well as for the absence of GFAP. The result is a product that is 85% pure population of postmitotic neurons comprised of both GABAergic and glutamatergic neurons without glia or any perforating cells. These neurons not only produce appropriate biochemical markers, but also display appropriate electrophysiological markers. Which can be captured by a patch clamp or multi-electrode arrays, which we will get into more in a bit. CDI's micelle neurons, produced in parallel fashion to our iCell neurons, can be generated engineered as well, which then allows us to make point mutations in places where clinical research has discovered disease-associated mutations. It is with this process that we can produce neurons that harbor known disease-associated point mutations, culture them up in a dish, and assess, utilizing many different approaches, the disease in a dish phenotype. The process of genetically engineering point mutations in the healthy donor background is only one process we can utilize to produce this disease in the dish. Here at CDI, we also investigated with success two other processes that result in phenotypic alterations, induced and innate. In the induced process, healthy donor human iPSCs are differentiated in different conditions, albeit medium conditions, or hypoxic conditions perhaps, and this differentiation process can produce phenotypic changes that can be assessed and compared to healthy controls. Another process, which Dr. Feaster will present on in a bit, is the innate, where we can take samples from a donor with a genetic disease and differentiate these iPSCs using the regular differentiation protocol and produce and compare them to the wild-type healthy controlled samples. Collectively, this is how CDI can leverage iPSC technology to advance personalized medicine. The neuronal disease models I will discuss today harbor mutations that result in terrible conditions that not only bad for the individual suffering from them, but the public healthcare system as well. Collectively, Alzheimer's, Parkinson's, and epilepsy disorders affect over 100 million people worldwide, and cost of these conditions in the U.S. alone is over $300 billion a year. This is obviously a need not only for disease understanding, but a major need for drug development for these conditions as well. One of the major hurdles within this drug discovery space is the lack of relevant cell models, which we believe here at CDI that we can start fulfilling with the disease in a dish approach. Alzheimer's disease currently affects the most amount of American individuals. It is a disease that is thought to result from plaque formations developing from aberrant amyloid beta and tau processes. These are both proteins that can be assessed via ready assay kits and things that we do regularly here at CDI. So as mentioned, the iCell neurons are cryopreserved and shipped to the customer. And once you guys have those in hand, you can plate them onto plates and then use regular assay kits to assess the amount of different protein levels. In this case, we're looking at amyloid beta, human tau, or human soluble APP alpha. And we can see over increasing number of cells in the culture conditions, we have increasing amounts of the protein. Furthermore, if we look and compare the amount of interesting amyloid beta protein levels, we can compare not only the 1 through 40 amyloid beta protein, but also the ratio of 40 to 42, which has been implicated in Alzheimer's disease. We can see with a gamma secretase inhibitor, we can see a dose-dependent decrease in the expression levels of these proteins, which matches what we would expect to see in the condition. We've generated multiple lines here at CDI that have interesting effects as far as Alzheimer's disease goes. So through research in 2012 in a Nature publication, there were 2 variants at the 673 location of the APP protein, and 2 different mutations can either, an alanine to a thiamine can be protective, or conversely, the A673V is causative. So this has been discovered and published on, and we here at CDI have put those in point mutations into our eye-cell neuron background, and we have discovered that the cells survive in culture, and thus we can actually investigate the levels of APP, or amyloid beta protein. Interestingly enough, we have discovered that the levels of 1 through 40 amyloid beta protein levels is decreased in the protective condition and increased in the causative condition, which is what we would expect. And then in neither of these cases has this disrupted the ability of the gamma secretase inhibitors to decrease the levels of these proteins of interest. If you put this onto the multi-electrode array platform, which is a platform to look at electrical activity of the cells, you can take these cells from the cryopreserved state, plate them onto these multi-electrode array platforms, and look at their activity levels, capture their waveforms, and capture their action potential time frames, and then describe these as far as a mean firing rate, and or look at interesting burst parameters, including in this case we looked at Poisson burst parameters on each individual channel. What we're showing you here on the left-hand side is firing rates in red and bursting rates in the lower blue for the wild type, the protective or the causative conditions for the Alzheimer's. And on the top, what we can see is over days in culture, the protective condition does not change the mean firing rate compared to the wild type. With that said, the causative condition is considerably lower in mean firing rate across the board and across all days in culture. The bursting rate on the bottom, we see at day 8 and day 15 that the protective condition here actually causes an increase in the Poisson bursting parameters, whereas the causative is decreased compared to control. Collectively, what this suggests is A, we have a protective and a causative sample for Alzheimer's disease here that we can make and have ready in cryopreserved vials. Furthermore, these can be assessed for the amyloid beta protein levels, and they display altered electrical activity levels. For the human Parkinson's model that I'll present in a moment, I'll first introduce the eye cell dopamine neurons, which again, similar to the eye cell neurons, is a neuronal product. However, in this case, it's a midbrain dopaminergic neuron that expresses over 90% co-expression for FOXA2 and LMX1, and over 80% for TH, which is tyrosine hydroxylase. These neurons express the appropriate markers compared to the human substantia nigra cells, and these cells, when placed in the multi-electrode array platform over days and cultures, beginning with day 6 and going all the way to 3 weeks in culture to day 20, they start to organize themselves and create large synchronous bursting behaviors. This can be captured in a functional assay to look at a velocity graph, which is an all-points histogram of the action potentials across all the electrodes. and we can see that there's hypersynchronous bursting behaviors that usually max out about 400, 500 hertz in their hypersynchronous moment. The engineered Parkinsonian line that we have is an alpha-synuclein A53T mutation, which is an autosomal dominant early onset Parkinson's mutation. This has been investigated with many publications. This is one of the most well-known Parkinsonian mutations. In the neurons that we have made this point mutation in display multiple attributes which would be in line with the idea that this is altering dopaminergic activity. The gene expression levels show that, first and foremost, there's an increased gene expression signal for alpha-synuclein, which is what we would expect in these neurons compared to control. Interestingly enough, the tyrosine hydroxylase and dopa decarboxylase measures are both decreased, suggesting that the production of dopamine in these neurons is actually lower than normal. And furthermore, the catabolism COMT gene expression level is actually higher than normal, suggesting that dopamine is getting degraded quicker as well. Both of these kind of push to the idea that there's less dopamine being organized within these dopa neurons, which is matching that which the research field sees. In the functional assay on the right-hand side with the velocity graph and the all-points histogram of synchronous bursting behaviors, what we can see two differences in the culture, which is with the same number of electrodes, we actually have decreased bursting rate, but during this hypersynchronous moment, we can actually see that the burst intensity is about 4 times more intense than the regular condition. Jumping into one of these publications a little bit deeper, what we can see is that in figure 2 of the paper from Namani, increased expression of alpha-synuclein reduces neurotransmitter release by inhibiting synaptic vesicle reclustering after exocytosis. In figure 2, it describes that it has decreased field EPSPs, which kind of matches the idea of our decreased bursting rate. But interestingly enough, it also has increased paired pulse facilitation, which suggests that during activity, you can actually induce more action potentials at once. This is also recapitulated in our functional MEA assay with the alpha-synuclein A53T dopamine neurons. The phenomena is further exemplified with the FDSS calcium assay system, which describes larger calcium oscillations and then earlier onset as far as day and culture goes. And then last but not least, the altered spare respiratory capacity that we see by assessment via seahorse mito stress test shows that there's altered levels of respiratory spare capacitance in these neurons. Collectively, the differential gene expression and the altered functional and respiratory capacitance measures of these cells is right in line with the idea that this is a Parkinsonian model and a good disease in the dish model to study this mutation. The epilepsy lines that we've investigated, and in this case I will present 3 different lines, is a KCNT1 mutation, an SCN1A mutation, and a GABA-A gamma-2R43Q mutation. These all confer different epilepsies in the clinic. All of these neurons can be plated the same as their eye cell neuron counterparts, controls, and they all show good growth and dendritic outgrowth. But one of the things that we've been able to capture using an IncuSight Zoom evaluation of neurite outgrowth is that the KCNT1 and SCN1A neurons both show increased neurite length and neurite outgrowth, neurite length and neurite branch points compared to their control conditions. This is suggestive of more dendritic outgrowth and more synaptic availability which could be a measure of epilepsy. And we also have done, obviously, functional measures with these neurons. After all, epilepsy is a brain electrical disturbance, and therefore measuring the electrical activity of these neurons is crucial. On the MEA, what we have discovered is if we capture the waveforms of the KCNT1 mutated neurons, we can see an increased repolarization and an increased after hyperpolarization, which is something that we would expect to find in this mutated neuron type. Furthermore, if we use an anti-epileptic agent, in this case published treatment of this condition was quinidine, we can see that actually quinidine attenuates the repolarization and after hyperpolarization increases we've seen with these waveforms. On the Poisson bursting parameter level, we can see that the KCNT1 neurons display increased intensity values and decreased burst durations compared to their wild-type counterparts. Again, this increased burst intensity level is a concept of an increased electrical signal suggesting an epileptic phenotype. The same can be said for the SCN1A neurons, which is also kind of more of a gas-on form of neuronal manipulation for epilepsy, whereas the R43Q neuron, which is a mutation in the GABA-A receptor and more like taking the brakes off version of epilepsy, we don't see an increase in the intensity value of the bursts, but what we do see is that we see twice as many excitatory bursts occurring. One cell type that I haven't mentioned yet that we do have is the eye cell glutamate neurons, which is a very excitatory version of the eye cell neurons, which synchronously bursts in culture. If we mix the eye cell neurons with the KCNT1 mutated epileptic neurons, what we can see is we see an increase in the synchronous bursting rate of the culture, something that, again, would be considered epileptic. And then if we treat this again with the quinidine condition in the same well, we can see that the bursting starts to ameliorate a little bit. So we're treating with AEDs and getting the decreased result that we would expect. So the highlights of the epileptic conditions we have is increased neurite outgrowth, epileptic functional phenotypes, and then again the treatment by the AEDs, which appears to be able to attenuate the condition slightly. I appreciate your attention, and then I will give this back over to the moderator, Robert, and then listen to TK Feaster's conversation. Thank you.
Thank you so very much, Doctor, for the beginning part of this presentation. And I would like to introduce Dr. TK Feaster for his portion. Dr. TK Feaster is a field application scientist at Cellular Dynamics International. Dr. Feaster received his PhD in pharmacology in 2015 from Vanderbilt University for his work using human-induced pluripotent stem cell-derived cardiomyocytes to investigate cardiac excitation contraction coupling. Dr. Feaster joined CDI as a technical application scientist working in the applications group, where he served as a primary support liaison for cardiac products, as well as sharing responsibility for in-lab applications development. I'll now turn it over to Dr. Feaster for his presentation.
Thank you for that introduction. Today I'm going to be talking about human iPS-derived disease models for hypertrophic cardiomyopathy. This is an agenda of the talk. First, I will go over some of the background on human-induced pluripotent stem cell-derived cardiomyocytes and introduce the eye cell cardiomyocytes. Next, I will talk about some of the disease modeling approaches employed at CDI, and then I will give you some background on hypertrophic cardiomyopathy, or HCM, really highlighting two key examples, one of an induced example and one of an innate example. Human-induced pluripotent stem cell-derived cardiomyocytes recapitulate many aspects of native cardiomyocyte biology. So these are human cardiomyocytes that give you access to human biology in vitro. Particularly, these cells display intact excitation-contraction coupling. This ranges from electrical activity at the membrane being translated to intracellular calcium handling with intact calcium-induced calcium release intact sarcoplasmic reticulum, and also intact calcium and myofilament interactions that drive mechanical beating and contractility. The I-cell cardiomyocytes are a cryopreserved cell that's ready to use. This is greater than 95% pure cardiomyocytes based on cardiac troponin T expression. These cells represent a consistent and reproducible source of human cardiomyocytes for in vitro assessment, including disease modeling, toxicity testing, and target validation. I'm just going to revisit this slide for a moment. Dr. Magnan went over some of the engineered examples for some of our neuronal disease models. I'll be focusing on the induced example and an innate example. In the induced example, the I-cell cardiomyocytes are exposed to different conditions to really bring out the HCM phenotype. In the innate example, the blood is taken from a patient with HCM that is reprogrammed to iPS cells. Those cells are then differentiated to cardiomyocytes and studied in the lab. For the innate example, I'll go into some of the characterization, the functional characterization of the mutation. And for the induced example, I'll give a high-level snapshot of the capabilities and the utility of this line. So what is HCM? HCM is classically characterized with preserved systolic function and impaired diastolic dysfunction. This affects 1 in 500 people and is accompanied by increasing occurrence of arrhythmic events and can lead to sudden cardiac death. HCM is characterized with thickening of the heart muscle. Here you can see in the diseased heart, the heart is thicker, particularly the septum, relative to the normal heart. HCM can be caused by many sarcomeric mutations. In particular, the beta-myosin heavy chain mutation accounts for over 50% of HCM cases. Mutations in this gene really have a severe phenotype. These can be studied in vitro by looking at the structure or morphology of the cells, looking at changes in cell size and reorganization, as well as changes in gene expression, increases in fetal gene expression, as well as an increased expression of beta-myosin heavy chain to the alpha-myosin heavy chain ratio. This can be assessed functionally by looking at changes in calcium handling, electrical activity, or contractile properties. So now I'm showing you one of the induced examples. These cells are plated as a monolayer and exposed to endothelin-1 to really bring out that cardiac hypertrophy phenotype. Here I'm showing the cells being stained with alpha-actinin to assess cell size. You can see that as you increase the concentration of endothelin-1, there's a concentration-dependent increase in the total cell area, showing that these cells are getting larger. We also looked at the F to G actin ratio to look at cytoskeletal rearrangement. Here we're showing the cells stained, and this is quantified below. As you increase the concentration of endothelin-1, there's a concentration-dependent increase in the FG actin ratio. We also looked at the increase in the fetal gene program, looking at BNP, or brain natriuretic peptide. We stained the cells with DAPI in blue and BNP in green, or antibody for BNP in green, and you can see that the induced cells have increased expression, and this is quantified below, where you have an increase, concentration-dependent increase in BNP expression per cell. So these data really show that you can use the I-cell cardiomyocytes and manipulate the environment to look at cardiac hypertrophy, looking for possibly compounds that will induce hypertrophy, compounds that might inhibit hypertrophy, or just really trying to understand the biology of how the cells are becoming hypertrophic. So next we're going to change gears and look at one of the examples of the innate hypertrophic cardiomyopathy. This is the micelle line that is derived from a patient with a R403Q mutation. This is a substitution that switches the arginine to glutamine. In panel A, I'm showing the control I-cell cardiomyocytes expressing greater than 95% cardiac troponin T. And we also have the disease cells that are comparable. These cells also have comparable viability as the control cells, as well as plating efficiency. In panel C, I'm showing a representative bright field image of the cells just to give you an idea of what they look like in culture. One of the first things we did was look at the BMP expression and the baseline BMP expression in the micelle 403Q cells. So you can see that there's increased BMP expression. This is done by staining the cells with the antibody for BMP. You can see that in red there's increased expression relative to the control cells. We also looked at some of the gene expression for this beta to alpha myosin heavy chain ratio. This is known to be increased in failing hearts or HCM hearts. And you can see that in vitro, these cells have increased expression, increased ratio of beta myosin to alpha myosin heavy chain ratio. So next we're going to look at a little more in depth at the gene profile. Here we're showing an example of the induced cardiac hypertrophy relative to control. And this is a panel of genes known to be involved in cardiac hypertrophy. And in panel B, I'm showing the R403Q sample relative to control for the same panel of genes. You can see that some of the genes are upregulated and some are downregulated in both cases. The trends are consistent across the induced and the innate samples. However, the induced sample has a greater increase or decrease relative to the control. So this is a more pronounced or robust phenotype relative to the R403Q samples. So the next thing we wanted to do was to look a little more closer at the functional consequences of this R403Q mutation. One of the first things we did was look at the beating activity, and this is assessed on the excelligence by looking at the changes in impedance. In these experiments, the cells are plated as a monolayer in an E plate. And in panel A, I'm showing the control I-cell cardiomyocytes representative impedance morphology. You can see that there's stable beating in these cells. In the R403Q cells, this beating is actually slowed. These cells are beating much slower than the control cells. This is quantified in table B. You can see that the R403Q cells beat at around 8 beats per minute, while the control cells in these conditions are beating at around 27 beats per minute. The amplitude of each cell type was the same. However, the beat rate variation was different. The R403Q cells had increased beat rate variation relative to the control cells. This was important because this is consistent with what you would see in vivo, that these cells or patients with this mutation have increased arrhythmic-like activity or unstable beating. Next, we wanted to look at the electrical activity. I've just showed you an example of the impedance looking at the beating behavior. This is now in more of a direct measure of electrical activity using the MEA. In these experiments, the cells are plated as a high concentration dot over the recording electrodes, and the electrical activity is measured. In panel A, I'm showing the control I-cell cardiomyocytes representative field potential morphology. And in B, I'm showing the R403Q mutation, the representative field potential duration. You can see that consistent with the impedance measurement, this beating is slowed relative to control. This is quantified in panel B, where we're looking at the field potential duration for the R403Q sample in green and the control cells in blue. You can see that the R403Q cells have a prolonged field potential duration relative to control cells. This is consistent with a lower beat frequency relative to the control cells, and these cells also display increased occurrence of beating irregularity or arrhythmic events, as shown in the beat rate variation. So this is just an independent experiment that really confirms the impedance experiment the same result as decreased beat rate and increased arrhythmic-like activity in the R403Q cells relative to control. Now we're going to change gears a little bit and look at some of the calcium handling properties of these cells. These are two publications that have shown using iPS-derived cardiomyocytes, they investigated beta-myosin heavy chain mutations. These mutations are different than the R403Q mutation. One is the R442G and the other is R663H mutation. Both of these mutations have a severe phenotype also. Lan et al. Showed in their paper in 2013 that the HCM sample that they looked at had a differential response to calcium. For example, they looked at nifedipine, which is a known calcium channel blocker, and they looked at the effect of nifedipine on beat rate, and they showed that as they increase the concentration of nifedipine, there's a concentration-dependent decrease in beat rate. However, with the HCM cells, this concentration response curve is shifted to the right, suggesting that these cells are less sensitive, that it takes more nifedipine to really stop the beating. This is consistent with the myofilament calcium sensitization hypothesis for HCM. We looked at this with the R403Q cells, and we saw the same response. We wanted to look a little more closer at the effect of calcium channel block on irregular beating as well as the effects of mechanical overload. Here we're using verapamil, which is another calcium channel blocker. In these experiments, we're looking at calcium handling by plating the cells in a monolayer and loading them with the calcium-sensitive dye. In panel A, I'm showing the calcium transients for the control cells. You can see the cells have stable, regular calcium transients. However, in the R403Q cells, these calcium transients are more irregular and slower, consistent with the electrical activity and the beating activity observed on the MEA as well as the impedance. These data are quantified in B, where you can see in green the control cells have a stable beating at around 30 beats per minute. However, the disease cells, looking at the calcium handling, have a reduced beat rate. They also display the irregular beating or increased arrhythmic activity. So we wanted to see what the effect would be of using verapamil, that calcium channel blocker, on these cells. In the control cells, verapamil has little effect on the beating behavior. However, in the R403Q cells, they restore the beating behavior and eliminate the irregular beating activity. This is quantified in D. You can see that in the green panels, the control cardiomyocytes as well as the verapamil control cardiomyocytes, there's negligible effects. However, when you expose the diseased R403Q cardiomyocytes to verapamil, the beat period is increased, nearly to baseline. And the irregular beating is reduced. The next thing we wanted to do was to look at the effects of mechanical overload. In these experiments, we're using the same calcium handling experiments, the same calcium handling dye, where we have the control cells and they're spontaneously beating. Then they undergo a pacing protocol where we increase the pacing to 0.5 hertz and then stop pacing and allow the cells to recover. Then increase the pacing to 1 hertz and stop pacing and allow the cells to recover. Then 1.5 hertz, stop pacing, and 2 hertz and stop pacing. You can see that at each event when the pacing is stopped, the cells begin to spontaneously beat again. However, when you look at the disease cells, the R403Q cells, at the lower frequencies of pacing, the cells recover when the pacing is stopped. However, when you increase to these higher frequencies, 1, 1.5, and 2 hertz, once you stop pacing, the cells do not recover. So this is a distinct phenotype that's shown in these cells. We looked at the effect of verapamil, and we showed that verapamil will reverse this effect and allow the R403Q cells to begin beating again spontaneously after pacing at these high frequencies is stopped. So this is an example of a unique phenotype to these cells that can be reversed with chemical inhibition of calcium channels. One could use this possibly to look at a phenotypic screen for compounds that will have a similar effect. This slide is to illustrate how we have been trying to really enable the entire research community to have access to different disease types through our banking efforts. The first iPSLs being banked in 2010, and then in 2015, approximately 800 donors being banked, and then currently approximately 2,000 donors. And the projection for 2018 is around 8,000. And these are across different disease backgrounds, from neurological disorders and cardiac disorders, of hypertrophic cardiomyopathy, dilated cardiomyopathy, and some neurological disorders. And some of them are disorders that affect the hepatocytes and different systems of the body. So this is the summary slide summarizing both Dr. Magnan's presentation and mine. Dr. Magnan went over some of the iPS-derived neuronal disease models for Alzheimer's, Parkinson's, and epilepsy, and these were all engineered examples. I went over some of the cardiac examples for hypertrophic cardiomyopathy, an induced example and an innate example, and then I also touched a little bit on some of the banking efforts to allow the research community access to these cell types. With that, I will pass it back over to the moderator, and thank you for your attention.
Well, thank you for that informative presentation, doctors. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. So we do have our first question here. Have you investigated other inducers of cardiac hypertrophy?
Yes, no, that's a good question. We have looked at other inducers to really induce and bring out that cardiac hypertrophic phenotype. We have looked at isoproterenol as well as phenylephrine, and all of those that we've tested have shown a response. However, the endothelin was the most robust response in our short assay. So some of the other inducers will work. However, the workflow needs to be adapted a little bit.
Well, thank you. Our next question, can this be applied to studying ME that affects over 1 million in U.S., perhaps 17 million worldwide, and is not FDA-approved diagnostics or treatments? Can you form natural killer cells, B cells, glial cell models, major process this year in ME, CFS, biochem, but need models?
That's a really good question. We can produce immune and glia cells from iPSCs, and all of these cells and the eye-cell neuron product itself are very competent for infection, unlike primary neurons, suggesting researchers would be able to produce an ME in a dish disease model from these samples.
Thank you. With your mutant protein expression experiments, are you also reducing the endogenous protein via RNA, Cas9, etc.?
The A53T mutation is in the genome and not overexpressed. So all the models that we have shown result from genomic differences and are an endogenous expression change for the mutant protein, not something that we've further manipulated as far as any type of up or down regulation goes.
Thank you. And do either speakers have examples involving a combination of both innate and induced states of, i.e., environmental insult onto cells from a diseased patient?
Yes, this is actually a very good question. I don't have any examples of this. However, that is clearly where the field is going, and the utility of these cells, that you can have the different disease backgrounds and then expose them to different environmental factors. So that's something that we definitely would like to look at, but I don't have any example of anything in the literature to reflect that.
Yeah. How likely is iPSC-derived disease model mimicking a real disease?
The idea of a model is that it's a model. It might not be exactly producing the phenotype that we see in the human whole body condition, but nonetheless, alterations in expression levels or changes in respiratory behavior or activity levels can be compared from the healthy control and then investigated in that fashion.
All right, thank you so very much. Looks like here might have time for one last question. Is there a correlation between firing frequency, respiratory capacity in general? Meaning, do neurons with lower frequency have higher respiratory capacity and the A53T neurons a disease-associated phenomenon?
A really good question, and obviously something that needs to be investigated more. Here's an example of does the model definitely recapitulate the human condition? In a lot of aspects of Parkinson's, we see in the human condition, in other models, we see a lowered spare respiratory capacity. In the cells that we've created with the A53T mutation, we actually see a significantly increased spare respiratory capacity. However, this is very early in culture, and as we know, Parkinson's is a longitudinal disorder. So maybe if we kept the cells in culture for a really extended period of time, similar to the disease condition, we could see. But nonetheless, the altered respiratory capacitance of the cells shows that there's definitely a phenotype and is worth investigation, and obviously something that we would like to look into in the future.
Well, thank you. Can you rescue any of you mutant phenotypes with overexpression?
So for this question, I don't have a precise answer for it, depending on the mutation and the disease. So I talked a little bit about the beta-myosin heavy chain mutation. I'm not sure that overexpressing that would rescue that phenotype. I don't think that's been looked at in the literature. One might try to reverse the mutation by genetically altering it, but that hasn't been done either.
No, thank you so very much for that. As far as final comments, doctors, do you have any final comments before we conclude today's presentation?
Obviously, it's a snapshot into the future. The personalized medicine space is in need of drug discovery and directional research, and using these models of the engineered and the innate can maybe give us a better glimpse into what's occurring with these disorders and diseases and allow us to enter that personalized medicine space with a little bit more direction and effort.
Well, thank you so very much. Just want to thank our sponsor for today, Cellular Dynamics, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.